Taste, or gustation, is a sense that develops through the interaction of dissolved molecules with taste buds. There are 5 basic tastes: sweet, salty, bitter, sour, and umami. Saltiness is mostly the taste of sodium. Sourness is mostly the taste of acid. Sweetness comes from the taste of sugars and certain proteins, and bitterness from potassium, magnesium, caffeine, etc. Umami is the most recent taste sensation described, and mostly comes from the taste of glutamine. Taste is associated mainly with the tongue, although there are taste receptors on the palate, and epiglottis as well. The surface of the tongue, along with the rest of the oral cavity, is lined by a stratified squamous epithelium. On the surface of the tongue are raised bumps called papillae that contain the taste buds. There are 3 types of papillae based on their appearance: velate, foliate, and fungiform. The number of taste buds within papillae varies, with each bud containing several specialized taste cells for the transduction of taste stimuli. These receptor cells release neurotransmitters when certain chemicals in ingested substances are carried to their surface in saliva. Neurotransmitters from the gustatory cells can activate the sensory neurons in the facial and glossopharyngeal cranial nerves. When a receptor cell is activated, its membrane potential changes, thus creating a receptor potential. Let's look into the mechanism of transduction for taste. First, for the taste of saltiness. On the taste receptor cells, there are amiloride-sensitive sodium channels. They are insensitive to voltage and stay open all the time. When the exterior concentration of sodium rises, sodium diffuses into the receptor cell, driven by the concentration gradient. This causes a depolarization, which in turn causes voltage-gated sodium and calcium channels to open near the synaptic vesicles, triggering the release of neurotransmitter onto the gustatory afferent axon. Now let's look into the taste of sourness. The taste of sourness is mediated by protons. Protons can permeate the amiloride-sensitive sodium channels, creating an inward proton current, which depolarizes the cell. Protons also bind and block potassium-selective channels, decreasing the permeability of the membrane to potassium, which also depolarizes the cell. As the concentration of protons increases because of ingesting acidic compounds, the depolarization of specific taste cells increases. The other 3 tastes, sweet, bitter, and umami, are transduced through G-protein-coupled cell surface receptors instead of the direct diffusion of ions like with salty and sour. The sweet taste is the sensitivity of taste cells to the presence of glucose dissolved in the saliva. Molecules that are similar in structure to glucose will have a similar effect on the sensation of sweetness. Other monosaccharides such as fructose or artificial sweeteners like aspartame, saccharin, or sucralose will activate the sweet receptors as well. The affinity for each of these molecules varies, and some will taste sweeter than glucose because they bind to the G-protein-coupled receptor differently. As for the bitter taste, the taste buds can be stimulated by a large number of molecules collectively known as alkaloids. And finally, for umami, like sweet and bitter taste, it is based on the activation of G-protein-coupled receptors, in this case by amino acids, especially glutamine. Once the taste cells are activated, they release neurotransmitters onto the dendrites of sensory neurons. These neurons are part of the facial and glossopharyngeal cranial nerves, as well as a component within the vagus nerve. Axons from the 3 cranial nerves carrying taste information travel to the medulla. From there, much of the information is carried to the thalamus, and then routed to the primary gustatory cortex, located near the inferior margin of the post-central gyrus. It is this primary gustatory cortex that is responsible for the sensations of taste. If you like this video, please hit the like button below, and subscribe to my channel to see more videos in future. Thank you for watching. Bye.